We're not recording. Let's do some attendance. Chief Brumfield, not here yet. Champion is here. Gorby, not here. Rose, I think, is online. Gunther is not here yet. Randall is here. Zion is not here yet. Keys is here. Jackson is here. Ez is here. Sigmund. Sigmund is here. Slaughter is not here yet. Smith is here. I thought. Yeah, back there. Stover's not here yet. Tompkins not here yet. Nest is here. Watson is here. Young is here. Flyback is here. Lawrence is here. Wilson is here. And Bill came in while I was doing attendance. Did I miss anybody else? But Harvey there. Belt's here. Oh, yeah. All right. Okay. Anybody else? All right, nice and easy. Let's get started. Announcements and reminders, as always, attendance is mandatory, rated. First word for attendance today is Charleston. Today is National States and Capitals Day, so since Charleston is our capital, that's the first word for attendance. As always, if you're here, you don't have to send those words in, but you can for extra credit as long as you send them in before 9 a.m. If you're online right now watching this, you do have to send those letters in, put those words in before 9 a.m. And of course, if you're watching the video at a later date, you take a screenshot every time I say it on the screen when they attend this meeting. So, any questions about that? Okay, just a reminder again, you can still retake exam 1. Take it as many times as you want. It's open book. It's not timed. Only catch it is you have to let me know when you're done, because I have no idea of knowing if you've done it. The grade will not automatically update. So let me know if you've retaken exam 1. Any questions about that? All right. We do have lab this week as usual. That's just a reminder. We'll start at 9 a.m. All it's going to be is a pre-lab lecture. I'm going to talk about next week's lab. I'm going to talk about how to write a lab report. And I'll do that. So all you need to do is be there. There's nothing to turn in. Being there on time. It's going to give you the full 20 points for the lab. Any questions about that? Okay. Do that is due on Friday as usual. It's the one on photosynthesis and respiration. If I remember correctly, there's 2 videos to watch. Then an intro. Then you have to answer questions about the introduction, and of course, a few hypothesis questions after that. So let me know if you need help with that. And then finally, just to let you know, I did take the weekend off. I didn't do any grading, didn't check any emails. So if you sent me anything over the weekend, or turned anything in and haven't heard back from me, that's fine. So I hope you get caught up today. That's it. Does anybody have any questions? Announcements? Like that? Okay. Nice and easy. So to remind you, what we're talking about now, there we go. We're about to talk about cellular respiration. And to remind you, exam 2 is mostly going to be about energy, right? We're talking about the different properties of life, that transformation of energy. This is what this exam is going to be mostly about. And also, exam 2 was mostly going to be about photosynthesis and respiration. There are going to be other questions, some of the other stuff we talked about enzymes and activation energy and all that stuff we talked about previously. But the large majority of the questions are going to be about photosynthesis and respiration. And those questions are almost entirely going to be choosing a particular stage. For example, I might say something like, in which stage is oxygen used in respiration? In which stage is water produced in respiration? So as I'm teaching this, keep that in mind. That's mostly how this exam is going to go. And also, like I said on Friday, everybody learns different ways and has their own method of studying. But my recommendation for these 2 chapters, or these 2 subjects, respiration and photosynthesis, is to memorize the big picture and then start adding details, right? So you should have this memorized, this slide right here. You should have this memorized before you start working on adding the details, right? So you need to know that respiration is broken down into 3 stages: glycolysis, citric acid cycle, and electron transport chain. Or electron transport. You need to know that. Then, once you have that memorized, of course I'm going to teach you all this anyway, but then once you have that memorized, then you can start adding the details, like what happens in glycolysis, what happens in the citric acid cycle, what happens in electron transport. So that's my recommendation. I also want to point out, you can see here, it says electron transport, in parentheses, oxidative phosphorylation. So that's the term that the book uses, oxidative phosphorylation. I call it electron transport, other people call it electron transport, and you'll see why. I actually do that to make things easier for you. It'll help you memorize some of this stuff. Likewise, citric acid cycle also goes by another name. It's also called the Krebs cycle with a K, but I call it the citric acid cycle for a reason, and you'll see why. It's going to help you memorize this stuff. So, any questions before we take this overview? We're going to have a big overview where we just look at the big picture, and then we'll start digging into the details. Any questions before we get started? All right. Let's look at the overview then. If you download the PowerPoint, you can watch this animation that shows cellular respiration in action. Here we go. The first stage is glycolysis. Like I said, you need to know that. Memorize that fact. Memorize that first stage is glycolysis. And basically, what happens in glycolysis, and again, this is an overview. We are going to come back to this and go into more detail. In glycolysis, glucose is split up into two molecules of something called pyruvate. This is another one of those things that has another name too. It's also called pyruvic acid. I call it pyruvate. Again, you'll see why. We get to make it easier. Now, your book says that the glycolysis usually occurs in the cytoplasm as opposed to in the mitochondria. This is the one part of the process that does not take place in the mitochondria. But it also says usually, right? So your book did put that term in there. But as far as we're concerned, as far as the exam is concerned, glycolysis happens in not in the mitochondria. It's the one stage that happens outside of mitochondria. That will be a test question. So there's a few test questions right here on the screen. What is the first step of respiration? Glycolysis. Which of the three stages uses glucose? Glycolysis. Which of the three stages produces pyruvate? Glycolysis. And which of the three stages happens in the cytoplasm or not in the mitochondria? The answer is glycolysis. So again, this is just an overview. We're going to come back to this and go into more detail. So I won't take questions yet. I'll take questions when we get back to it. The second stage is the citric acid cycle. Again, it's also known as the Krebs cycle, but I call it the citric acid cycle for a reason. This one does happen in the mitochondria. And this is the part that completely breaks down glucose. So if you remember from the previous slide, you start with glucose at glycolysis, and you break that down into something called pyruvate. And then that pyruvate goes into the citric acid cycle and is broken down even more. And in the process of doing that, it produces carbon dioxide. So again, test questions on this slide. What is the second stage of respiration? Citric acid cycle. Which of the three stages happens inside the mitochondria? Citric acid cycle, and the one you're about to learn. Which of the three stages produces carbon dioxide? Citric acid cycle. That reminds me. Now I can tell you why I call it the citric acid cycle instead of the Krebs cycle. Anytime you get a question on this exam that says anything about carbon dioxide, right? Whether it's producing carbon dioxide, like here in the citric acid cycle, or later when we talk about photosynthesis that uses carbon dioxide, either way, when you get a question about carbon dioxide, the answer is going to be some sort of cycle, whether it's the citric acid cycle or the cycle that you're going to learn later for photosynthesis. So again, that's why I call it the citric acid cycle. All those C's hopefully will remind you that that's the one that produces carbon dioxide. Citric acid cycle C, carbon dioxide. And again, it's just an overview. We're going to come back to this and talk about it in more detail. So that brings us to the next stage. Actually, before we even talk about the next stage, it's worth noting that the first two stages do generate a small amount of ATP. And hopefully you remember from last week, I said that's the one thing you should memorize before you memorize anything else from this chapter, right? Your cells need ATP, right? That's where they get their energy from. So this whole thing, this whole discussion, basically the whole exam is all about how do we get ATP. So again, glycolysis and citric acid cycle, you make a little bit of ATP. More importantly, they make this thing called NAD+, which I'm going to explain later. Again, this is just an overview, this is an introduction, we're going to get into more detail later. But the main purpose of glycolysis in the citric acid cycle is to strip away high energy electrons. The real moneymaker in this process is the third step, which is the electron transfer, and the electron transport chain, oxidative phosphorylation. That's the third stage, and it's the one that makes the most ATP. So in a sense, it's these first two stages that are just there to support the third stage, right? It's all about stripping away these high energy electrons that are going to be used in the electron transport chain. This is what we're about to talk about. And again, this is just an overview. We'll come back and talk about it in more detail later. So electron transport chain, again, aka oxidative phosphorylation. On the exam, it's probably going to be called the electron transport chain, because again, I'm hoping this will make it easier for you to remember. This whole process is all about stripping away high energy electrons, right? That's what the first two stages do. If you can remember that the third stage is the one that makes the most ATP, hopefully the name electron transport chain will help you remember that's where the electrons are going, right? We're stripping away electrons. They're going to be taken somewhere. Where are they being taken to? The electron transport chain. So what happens is we have all these high energy electrons that have been stripped away. They come to the electron transport chain, and slowly but surely they're kind of stripped of their energy. So again, I keep saying high energy electrons, right? Not just electrons, but high energy electrons. They can't stay that way for a long time. It's unstable. So as they kind of lose that energy, right? As you learned from last week, I think it was, you can't destroy energy, right? We can only convert it from one form to another. So when these high energy electrons release that energy, it disappeared, right? It's of use. And what happens is there's these protons inside the inner membrane of the mitochondria, and I'll explain that later in more detail. But what happens is the electrons go from one protein to the other. And then the process of doing that, again, they're releasing that energy. That energy is used to pump hydrogen ions against their concentration gradient. We'll talk about that in more detail. So that's the big picture. Again, we're going to go back and talk about all three stages individually. But that's how I would recommend studying it, right? Study the big picture first, have that memorized, and then worry about the details. Speaking of the big picture, you're right. If you take notes and you think I'm going too fast, I apologize, because again, we're going to come back to this. So if you're taking notes, you can give your hand a break. But also, this is another thing you should memorize. Memorize the equation, right? So we have one molecule of glucose, 6 molecules of oxygen, gives us 6 molecules of carbon dioxide, 6 molecules of water, and about 38 ATP. So make sure you have that memorized. That's the overall equation. And again, there's going to be a lot of questions about this. Like, which of the 3 stages uses glucose? Glycolysis. Which of the 3 stages uses oxygen? Electron transport chain. I haven't talked to that yet, but we'll talk about it. Which of the 3 stages produces carbon dioxide? Citric acid cycle, right? All those C's. Which of the 3 stages produces water? Electron transport chain. I haven't talked to that yet either, but we'll get there. Which of the 3 stages produces ATP? All 3 of them. Which of the 3 stages produces the most ATP? Electron transport chain. So again, this is a big overview. Here's a big overview picture. This is from the old textbook, not from your textbook. As you can see here, we're looking at a cell. There's the mitochondria. Like I said earlier, glycolysis is the only one that happens outside of the mitochondria. The other 2 happen inside the mitochondria. Glycolysis takes glucose, breaks it down into pyruvic acid, and produces a little bit of ATP in the process. That pyruvic acid goes through some changes, then goes into the mitochondria, into the citric acid cycle, where it's breaking down all the way. It's produced carbon dioxide, which is not shown on this picture, produces a little bit of ATP. But again, like I said earlier, more importantly, these 2 stages, as they're breaking down this glucose, stripping away high energy electrons. Those high energy electrons go into the electron transport chain. That's where the majority of the ATP. So that's it. That's the overview. So now we can get into it and start looking at more details. So any questions before we get started? All right, here we go. So stage 1, glycolysis. So this is basically what I taught you earlier. It's just now we're going to have more details to it. As you should already know, because you've drawn this in lab, glucose is a 6-carbon molecule. And what happens during glycolysis is basically it's broken in half. So obviously, if you have something that's 6 carbons and you break that thing in half, then you're going to be left with 2 new molecules, each of which are going to have 3 carbons. And that is called pyruvate. So you need to know this. Again, the overview, I just said we broke down glucose to make pyruvate. Now I'm giving you more details. I'm reminding you that glucose is 6 carbons, right? And it's broken down in half, which again leaves us to 2 pyruvate molecules, each with 3 carbons. Any questions about that first bullet point? Okay. Again, more importantly, we're stripping away high energy electrons in this process. So now I'm going to explain what this molecule is. This NAD+, plus, you can almost think of it just like it's a bucket. It's a bucket that carries electrons. Technically, it carries 2 electrons. Each molecule carries 2 electrons. I'm not going to get into that much detail. But that thing, you see it's got plus next to it, right? That means, obviously, it's going to be attracted to negative things. And what are our electrons? They're negative. So this thing is an empty bucket, so to speak. NAD plus is an empty bucket ready to carry some electrons. Once it gets the electrons, technically it has a different name, and technically that's called NADH. I'm probably not going to get into that much detail on the exam. So for you, if you see any molecule that looks like this, you should think, yeah, that's the thing that's carrying electrons. And yes, technically, if it has a plus next to it, that means it hasn't gotten the electron yet, right? It's still an empty bucket. And technically, if it has an H at the end, that means it has the electron. But again, I don't think it's going to be that detailed on the exam. I just want you to associate anything that looks like that with moving electrons. And you're going to see throughout this chapter and the next one, there's also going to be 2 other molecules that basically move electrons around. And their names, we'll get to it, but they look very similar. So again, when you see anything that looks like that, you should associate it with carrying electrons. And I'm not going to ask you to differentiate between this NADH and the other ones I'm about to teach you. You're not going to need to know the difference. See anything that looks like that, you should think those are the things that are carrying around electrons. So again, during glycolysis, we're breaking down glucose to make pyruvate. In the process of doing that, we're stripping away these high energy electrons, but also we do make a little bit of ATP. Specifically, 2 molecules basically. And I'm not even going to get that specific on the exam. Like I said, there's going to be a test question that says, which of the 3 stages produces ATP? And the answer is all 3 stages produce ATP. Then there'll be a test question that says, which one produces the most ATP? And that's the electron transport chain. Again, glycolysis does produce a little bit of ATP, specifically 2 molecules of it, but I'm not going to get that detail on the exam. Anyway, this stage itself is also broken down into 2 stages. There's the energy investment phase and the energy harvest phase. And I'll show you a picture of that. And then once I do, I'll take some pictures of it. Again, just as a reminder with this picture, glycolysis is the only stage that happens outside of mitochondria. Products of glycolysis, which is pyruvate, goes through some changes and goes into the citric acid cycle. A little bit of ATP is made. More importantly, even though it's not really shown on this picture, what we've done here is stripped away high energy electrons that are then carried by that NADH to the electron transport chain. So anyway, like I said, there's 2 phases here. First phase is the energy investment phase, and the second phase is the energy harvest phase. So what we're looking at here, well, this is big. That's the whole overview. So this goes back to the slide I was talking about earlier, right? I said we have a 6-carbon molecule, 6-carbon glucose. It's broken in half. That gives us 2 3-carbon molecules, right? 2 molecules of pyruvate. So there's the big picture of glycolysis. Again, you already saw it in writing on the other slide. Now you're just seeing a picture. 6 carbons. Well, we still have 6 carbons, right? So now they're broken in half. So we have 2 3-carbon molecules. Anyway, energy investment phase. So like we mentioned earlier, when we were talking about enzymes, I said every chemical reaction has to have energy to start it off, right? Even a chemical reaction that's going to release a lot of energy, it still needs an input of energy. Sort of like even like a nuclear bomb, right? It's a huge amount of energy being released, but it still needs an input of energy to get that explosion started. Same here, right? We're going to produce energy, but before we do, we have to invest a little bit. Technically, we start off with 2 ATP. I'm not going to get that detailed into the exam, so you don't need to know that it's 2 of them. But you do need to know that before this process gets started, we do need to invest some energy. And of course, as you guys know, the energy is going to be ATP. Like I said last week, there are other forms of energy that we don't talk about in this course, which we look at later. As far as we're concerned, as far as the exam is concerned, it's all about ATP. And of course, like you learned last week, when ATP is releasing its energy, the way it does it is by removing one of its phosphate groups, right? So we start off with ATP, technically that leaves us with ADP, right? We also have a phosphate group, so now we have ATP. But again, I'm not going to get that detailed. And it's just a given. Anytime ATP is used, you end up with ADP. That's just the ATP cycle. But again, energy investment phase will require ATP. Now, for extra credit, you can look this up. Because I didn't tell you why. What do we even need that for? Actually, no, that's not true. I'll explain it a little bit. But I'm not going to go into much detail. You can look it up for extra credit. Again, this is the energy investment phase. What we've done is we transferred the phosphates from the ATP to that pyruvate molecule, right? So now each one of those 2 molecules has its own phosphate group on it. That brings us to the energy investment phase, which is what you see right here. So here, we've stripped away these high energy electrons, right? And we've added another phosphate. So now those molecules have 2 phosphates. And another thing I haven't told you, this is a good reminder. Last week I told you every single chemical reaction that happens in your cells has a specific enzyme for it. You notice I haven't told you any of that here. So you can look that up for extra credit if you want as well. What is the enzyme that takes the phosphate from the ATP and transfers it to this molecule? And also, what's the name of that molecule? Because technically it's not pyruvate anymore, but it has a phosphate group on it. Then, likewise, what is the enzyme that takes another phosphate and adds it to this molecule? Then what's the name of that molecule, right? So it's now technically a different molecule, because now we stripped away electrons and we added another phosphate group that technically has a different name. Don't need to know it for the exam, but look it up for extra credit if you want. So anyway, we have these extra phosphate groups attached to those molecules. So now we can strip those away, right? So we can use those phosphate groups to basically recharge our battery, right? So I told you ATP is basically like a dead battery. An ATP is the charged battery, right? So that's what we've done here. We've taken that phosphate group and put it back onto the ATP, which produces ATP. And you can see here, if you're good at math, it's kind of a joke, you don't have to be good at math to see this. We've produced 4 ATP, right? But of course, we already had to spin 2. So technically, like I said in the previous slide, we have a net gain of 2 ATP from this process. So even though I'm talking about it, I'll also remind you, I'm not going to ask you that on the exam. You do need to know it produces a little bit of ATP, but I'm not going to ask you the name or the number. And I'm explaining the number, right? We produced 4, but we spin 2, so we netted 2. Anyway, any questions about this slide? Okay, the most important thing here, this is a lot of information. But really, on the exam, there's not that much information from here that's going to be on the exam. The important thing here is that, we started off with a 6-carbon molecule, broke it down. In the process of breaking it down, we stripped away high energy electrons and produced a little bit of ATP. That's it. That's all you need to know. So any questions? All right, like I mentioned earlier, there's an enzyme for each one of these steps. It has its own specific enzyme. I'm not going to ask you about it. I'm not going to teach you. And this is a picture from the old textbook that actually shows, hey, look, there's an enzyme taking off this phosphate from that molecule and adding it to the ADP to make ATP. But That's from the old textbook. Your textbook doesn't have that. So again, once again, I'll remind you, look that up for extra credit if you want. What is the name of that enzyme? What is the name of this molecule? What is the name of this molecule? None of it is important as far as the exam is concerned, but if you want to look it up for extra credit. So any questions about step one? We're done talking about glycolysis. Right, so again, we started off with glucose, we broke it down into something called pyruvate. In the process, we produced a little bit of ATP, but most importantly, we stripped away high energy electrons. So let's talk about the next stage, citric acid cycle. Actually, for some weird reason, your textbook puts them together. If I were writing this textbook, I would have 4.3 would be citric acid cycle, and 4.4 would be oxidative phosphorylation. But I didn't write the book, so here we go. Let's talk about the learning objectives for this section. By the end of it, you should be able to describe the location of the citric acid cycle, that's the mitochondria, and oxidative phosphorylation, also the mitochondria. You should be able to describe the overall outcome of the citric acid cycle, strip away high energy electrons and produce carbon dioxide. There you go. And the overall outcome of oxidative phosphorylation, aka the citric acid, electron transport chain. And you should be able to describe the relationship of glycolysis, citric acid cycle, and electron transport chain in terms of their inputs and outputs. There we go. Let's talk about it. Now, this does get a little bit more complicated. Hopefully by now I've said it enough. You remember, product of glycolysis is pyruvate. Right? We took a 6-carbon molecule, broke it down, and we produced pyruvate. However, pyruvate cannot get into the mitochondria. We won't get into that either. You can look that up for extra credit. Why? Why can't pyruvate get into the citric acid cycle? Look it up if you want. The important thing here is you know that it can't. So now it has to be groomed. It has to be converted. So let's talk about that. Again, pyruvate, 3-carbon molecule. So the first thing we're going to do with this step to groom it to get it ready to go into the citric acid cycle is to lose one of those carbons. And in doing that, we produce carbon dioxide. So that's not new information. I told you that during the overview, but I'm telling it to you again. Which of the 3 stages produces carbon dioxide? The citric acid cycle, all those C's. Now, that will be a test question, which of the 3 stages produces carbon dioxide. But I'm not going to ask you is where that came from. Like I'm not going to be so specific where you need to memorize the fact that, all right, we had a 3-carbon pyruvate, and it lost a carbon, and that's where the carbon dioxide came from. Even though that's what I just told you, I'm not going to test you on that. What I want you to know is you produce carbon dioxide from the citric acid cycle. And also just memorize the fact that pyruvate is the product of glycolysis, that it cannot go into the citric acid cycle. So we have to make some adjustments to it. So technically, again, once you change the molecule, if you lose an atom or add electrons or take away electrons, it's technically a different molecule, right? So when we take that carbon away from pyruvate, it's no longer pyruvate. It's now called acetic acid. Now, so we'll start with. But yeah, your textbook doesn't talk about that. I might ask that question on the exam. I might say, which of the 3 stages is associated with acetic acid? The answer is citric acid cycle. So if you're taking notes, you might write that down. Because this bullet point is not in your textbook. One of those few times where I might test you on something that's not in your textbook. But here's the good news. It's easy to remember. Because, again, like I told you earlier, the citric acid cycle is also called the Krebs cycle. But I don't call it the Krebs cycle. I told you one of the reasons why I don't call it the Krebs cycle, because all those C's, I want those C's to remind you of carbon dioxide, right? Another reason I don't call it the Krebs cycle. Any question you get on the exam that has anything to do with acid, you should think of the citric acid cycle, even though we're talking about acidic acid, right, so far. We haven't talked about citric acid yet, but anytime you get a question that says anything about acid, you should think of the citric acid cycle. I did that on purpose. Just like when we talked earlier about pyruvate, right? Another word for pyruvate is pyruvic acid. I'm not calling it pyruvic acid because I'm telling you now, if there's any questions that ask about acid, you should think of the citric acid cycle. So I'm not going to mention pyruvate by its other name on the exam because that would be pyruvic acid, and that's not associated with citric acid cycle, right? So again, anything that has to do with acid, you should think of the citric acid cycle. Any questions so far? All right, let's see if anybody can guess this. In the process of taking away one of those carbons, what else do you think we did? Remember I told you this is the big picture. This is what the first two stages are all about. It's doing this thing that I'm about to tell you what I told you before. It happened in glycolysis. It's happening here. I just haven't told you yet. What else do you think is happening when we're doing this? Not only are we taking away that carbon, but we're taking away some other stuff. Somebody remember? Anybody guess? Okay. Electrons, right? We're stripping away high energy electrons. This whole thing is about stripping away high energy electrons. And yes, technically, we get all technical and say, you know, it's the NAD plus that's taking the electrons. Once it accepts the electrons, it's called NADH. Once again, I'm not going to get that detailed on the exam. What you should know for the exam, again, when you see anything that looks like this, NAD plus, NADH, other ones I'm going to teach you, see anything like that, you should just think, oh yeah, that's the thing that's stripping away the electrons. It's transferring the electrons from one place to another. Any questions so far? All right. Each acidic acid is then attached to another molecule called coenzyme A, which is abbreviated CoA. And once that happens, obviously again, when you make changes to a molecule, you also change its name. So now technically we have something called acetyl-CoA. You need to know that word for the exam, but I'm not going to get that detailed into I don't need you to memorize the fact that took pyruvate, took away carbon, which left us with acidic acid, and then we attach that acidic acid to acetyl-CoA, or excuse me, coenzyme A, making acetyl-CoA. I'm not going to get that detail. But there will be a test question that says which of the three stages is associated with acetyl-CoA, or maybe even coenzyme A. That's what you need to know, right? That is associated with the citric acid cycle. You do need to know. Now, I don't know, maybe I'll have some extra credit questions that make you recall all this. That we started off with 3-carbon pyruvate, we lost one, and then we had acidic acid. We attached that to coenzyme A to make acetyl-CoA. And I might maybe give you an extra credit question like that, but again, for the regular credit questions, you don't need to memorize all these details. You should just associate these molecules with that stage. So, any questions about that? Nice and easy, right? One thing I didn't mention, you can see it in this picture, is again, we're producing a little bit of ATP, right? So, we took the products of glycolysis, which is pyruvate, made some changes to it. In the process, we produced a little bit of carbon dioxide. That's going into the citric acid cycle. More importantly, we're stripping away high energy electrons, and we're also producing a little bit of ATP. We're not done talking about it yet. It's just an image to show you an overview of what we're talking about. Here's a good picture, I think. This is from your textbook. It might be from the old textbook, I don't remember. But anyway, this is the picture of everything we said, right? So we have this pyruvic acid. See, I didn't make that. I would call it pyruvate. We have this pyruvate for glycolysis, right? We lose the carbon to carbon dioxide, which leaves us with acidic acid. We're stripping away high energy electrons. We also attach it to coenzyme A, and that leaves us with acetyl-CoA. Pretty simple stuff. And again, most importantly, as far as we're concerned, stripped away those high energy electrons. That is going to be used in the third part, the third stage, which produces most of the ATP. Any questions about that picture? All right, the next word for attendance, methyl acid. That is the capital of the last state I looked at. So there we go. Again, we had that acidic acid. We attached it to the coenzyme A to make acetyl-CoA so we can get into the mitochondria. Once it gets into the mitochondria, basically we lose the acetyl-CoA. You can look up that for extra credit too if you want. Like, how does that happen? Where does the acetyl-CoA go? Also, why does that even need to happen? All the stuff that you don't need to know for this exam, which you can look up if you want. But anyway, now we're back to acidic acid, right? We're back to that 2-carbon molecule. Now it's broken down even more, right? So we're taking that 2-carbon molecule and breaking it down again, which is going to produce even more carbon dioxide, specifically 2 molecules of it. But put it next to there to remind you, visual reminder. I'm not going to get that detailed and ask you how many. The idea here is of the 3 stages of respiration, which of the 3 stages produces carbon dioxide? The answer is citric acid. Yeah. Okay. So again, in doing this, we are producing a little bit of ATP. But more importantly, like I said in the previous slide, now you can see it right. More importantly, we are stripping away high energy electrons. And here we go. Here's another one now. FADH2. Now you've learned yet another electron carrier. But like I said, don't those 2 things look the same? Like NADH, FADH2, they look very similar, right? As far as you're concerned, they are the same thing. They are technically different, but as far as you're concerned, they're just things that are moving around electrons. So like I said, there's going to be 3 of them. So eventually I'm going to teach you a 3rd one. For the exam, I'm never going to have you differentiate between the 3. You just need to think of those as the electron carriers. Anyway, all the carbons that have entered the cycle as fuel, right, so it's acetic acid, is now accounted for as carbon dioxide, and yet another molecule, a 4-carbon acceptor molecule, I'm about to show you on the next slide. But for the exam, you do not need to know about that 4-carbon acceptor molecule. Matter of fact, I don't even give you the name of it. Your textbook doesn't name it, so neither am I. You can look it up again for extra credit if you want. What is the name of this molecule? So you can see here, again, this is the cycle. We have this acceptor molecule. It accepts these 2-carbon acetic acid, which is what gives us citric acid. This is a 6-carbon molecule. That is broken down to an acceptor molecule, and again, in the process, where obviously if you're going from 6 carbons to 4, you're obviously losing 2 carbons, right? So those 2 carbons are gone as ATP. We're also producing a little bit of ATP. More importantly, we're stripping away high energy electrons. So again, we're really just looking at the big picture here. There's a lot of details we've left out in this 100 level course, and you can look them up if you want, right? So what's the name of this acceptor molecule? What are the enzymes responsible for each of these reactions? And also, just so you know, there's more reactions than just the 2, but yeah, this is an easy version, so I don't give you all the in-between stuff. But you can look it up if you want. Like there's more steps between here and here. Right? We don't just go from here to here magically. There's other things that I don't teach you about. Same here. Going from here to here, there's other steps involved. But you don't have to know them, but you can look them up for extra credit if you want. So that's it for the citric acid cycle. Does anybody have any questions about the citric acid cycle? All right, let's talk about the last one. So, like I keep saying, it's really important to remember the fact that we're stripping away high energy electrons. So, in a sense, this thing is all about electrons. Like we're stripping away high energy electrons in the first 2 phases, or in the first 2 phases. Those are taken to the last phase. That's where they're going to be put to use. Your textbook describes it as electrons falling down in a cascade, right? Like falling down stairs, releasing energy. Again, your cells putting that energy to use. So I haven't told you how yet. Obviously, you know the end game, right? You know at the very least, this is the first thing you need to know. Obviously, we're making ATP here, a lot of ATP. So you just don't know how yet. I'm going to teach you how. How do we turn all these high energy electrons into ATP? But before we talk about that, let's talk about some basic electronics. I don't know if any of you guys are familiar with electronics. And in a very basic way of stating it, electrons have to come from somewhere and they have to go somewhere. So that's why I've got this picture of these jumper cables, right? And if you only put one of the jumper cables on, you either don't have electrons coming in, right? Because you just put it on, depending on which one you put it on, or you don't have anywhere for the electrons to go. This is why you have to plug them both in, right? Clamp them both on, because the electrons have to come from somewhere and they have to go somewhere. If the electrons don't have anywhere to go, they won't flow, right? That's how electronics works. That's also how your cell works too. So we're stripping away all these high energy electrons from these molecules, but they have somewhere to go. If they didn't have somewhere to go, we would have an issue. We'll talk about that later too. But yeah, So this is what we're talking about, right? Electrons coming from somewhere, going somewhere. And now that we're done talking about the first two stages, I'll go ahead and tell you that is going to be one of the test questions. I think I may have already mentioned this, but which of the three stages strips away high energy electrons? Citric acid cycle, glycolysis. Which of the three stages produces or uses high energy electrons? The one we're about to talk about, electron transport chain. Another test question, I don't think I mentioned this, but I might ask, where do these high energy electrons come from? And as far as we're concerned, and there are exceptions, we'll talk about that later. As far as we're concerned, it's all about glucose, right? We're talking about breaking down glucose. Those high energy electrons came from glucose. That will be a test question. Glucose is giving those high energy electrons. Of course, technically, glucose is then broken down to pyruvate, which is then broken down into acetic acid, which is then transferred and turned into acetyl-CoA, whatever. But ultimately, that was glucose, right? We started with glucose, keep breaking it down into everything. But that's where the electrons are coming from. They're coming from glucose. That will be a test question. So, any questions so far? All right, that was nice and easy. So as a reminder, this is also a stage that's happening inside the mitochondria, along with citric acid cycle. Again, that will be a test question. Then later, we're going to talk about the fact that it's in mitochondria. Now, when we talked about the citric acid cycle, I said, yeah, that happens in the mitochondria. But I left it at that. For the electron transport chain, the mitochondria is actually a very important part of it. We'll talk about why. Now, again, just memorize the fact that that's one of the two stages that are in mitochondria. So again, we're talking about these high energy electrons, right? We've stripped away these high energy electrons. So the overall electron transport is like a downward trip for the electrons. They're giving off this energy to theirs. Again, this is a good test question. We're talking about where the electrons are coming from, where are they going. Again, like I said previously, they're coming through glucose, right? We're stripping them away using NAD+. Also, the other one I taught you, and another one that I'm going to teach you later, but it doesn't matter. They're all the same as far as we're concerned. We're stripping away these high energy electrons. Those high energy electrons are taken to the electron transport chain, which is what you're about to learn about. Here's a new piece of information. Oxygen is accepting those electrons. So, like I said, you need to know where the electrons came from. You need to know where they go. So that's the second part of that. That will be a test question. Where do the electrons go? Go to oxygen. Oxygen accepts the electrons. Also, in the process of doing that, it becomes water. Obviously, other stuff has to happen too. You can't just add electrons to water or add electrons to oxygen to get water. There are some other things happening, but we don't worry about those details. The point is, electrons come from glucose, oxygen accepts them, and that produces water. That will be a test question. I might should take this part off. I mean, this is true. Technically, the NAD+, obviously, is the molecule that's stripping away the electrons. But I don't want you to confuse you, because for the exam, there's going to be a question that says, the electrons come from blank, and they are accepted by blank, and form a molecule of blank. So there will be a test question that has those three blanks in it, and you need to know that. Electrons come from glucose, they're accepted by oxygen, and in the process of doing that, it forms water. That's one of the most commonly missed questions on the exam. Now, there'll be other test questions that you'll probably get right, like which of the three stages uses high energy electrons? Electron transport chain. Which of the three stages uses oxygen? Electron transport chain. Which of the three stages produces water? Electron transport chain. Any questions about this slide before I show you like a little visualization of all these words? Okay, here we go. So everything we said, but now let's just visualize, right? So we stripped away these high energy electrons from food, glucose as far as we're concerned. I'll bring that up later. There are exceptions. We take these high energy electrons, technically two of them for each molecule, but we don't get into that much detail. They go through the electron transport chain, literally proteins that I'm going to talk about later. It goes from one protein to the next. It releases energy, and that energy is used to produce ATP. This is going to be a whole discussion too. You're going to have to learn how that energy is used to make ATP. We'll get there. All those electrons are accepted by a molecule of oxygen, which then produces water. And this picture is not from your textbook. And you can see there are some more details. Like I said, technically this is 2 electrons, but I'm not going to get into that much detail. Technically we have half a molecule of O2. I'm not going to get into that much detail. Technically, like I said earlier, you can't just add electrons to oxygen and get water, right? You also need hydrogens, so that comes from there too. But again, we don't get into that much detail. The idea here is we strip away energy, strip away high energy electrons with glucose, and it's ultimately accepted by oxygen, which produces water. So any questions? All right, so let's talk about this. Like I keep saying, these high energy electrons, they go from protein to protein. In the process of doing that, they're releasing energy and producing ATP. That's a little bit vague, so let's talk about how that happens. So the proteins of the electron transport chain are built into the inner membrane of the mitochondria. This will make a lot more sense on the next slide when I show you a picture. But to remind you, the mitochondria has 2 membranes. There's an outer membrane, and there's an inner membrane. The inner membrane is very highly folded. Like imagine if you had like a Kroger bag, a small plastic bag. Then imagine you had a huge trash bag. You crumbled it up, stuffed it inside the Kroger bag. That's sort of what you have here, right? You have the outer membrane, which is not that folded, but the inner membrane is a lot bigger, so it's kind of squished together. And it's highly folded. And that is not trivial. Obviously, I wouldn't be teaching it to you if it was trivial. This is actually important. Here we have another structure-function relationship. The fact that it's highly folded helps it do its job. Because, basically, it has a large surface area. And obviously, if we're the thing doing the work of these proteins embedded in the membrane, obviously the more membrane you have, the more proteins you have, therefore, the more ATP. So anyway, let me show you the next slide before I take pictures. This will make a lot more sense. So that's what the mitochondria looks like, right? We've kind of like cut into it. So here's the outer membrane. We've cut into it, so now we can see here's the inner membrane. Then they cut into that, so you can see like the actual inside of the mitochondria. That's a drawing of it. This is what it actually looks like. This is the electron microscope picture of the mitochondria. But yeah, that's how it works. Now notice there's some words here. You don't need to know these words. Cristae, matrix. You don't need to know any of that. I'm not going to use those words. I'm not going to test you on those words. But again, the fact that it's highly folded is important, so there might be a test question on that. Also, the fact that we have an inner membrane and an outer membrane, that will also be important coming up. I haven't told you why yet, but there is an important thing about this that you're about to learn. So any questions so far? Okay, so think about what you learned about diffusion, right? When we talked about diffusion and potential energy, right? So this is a good example. When you think of a dam, right, you have all this water dammed up. That's kind of a bad picture, but on the other side of that dam is like basically a lake, right? That dam is keeping the water from coming in this way. Gravity, right? Nature wants the water to come this way, but the dam is holding it back. So there's a lot of potential energy behind that dam. And also, side note, this is how we sometimes make electricity, right? We can open that up and let the water flow because nature is going to want to make the water flow. In the process of doing that, we can turn a turbine and make electricity. And in a sense, this is also what's happening in your cells. So it's not water we're dealing with. What we're dealing with is hydrogen ions. So you need to know that for the example as well. So I keep saying, strip away these hydrogen electrons, take them to the electron transport chain. They go from protein to protein, releasing their energy to produce ATP, but I never mentioned how. This is the how. So as those electrons go from protein to protein, they're releasing their energy. That energy is used to pump hydrogen ions against their concentration gradient. So again, go back to what you learned about diffusion. So what we're talking about here is active transport, right? We have to use energy because it goes against nature, right? We're going against the concentration gradient. Nature wants to diffuse things, right? Nature wants things to go down the concentration gradient. But here we're going against the concentration gradient. Now finally you understand where that energy is used, right? I keep saying we strip away these high energy electrons. Those electrons are putting off their energy. Now finally you know what it was used for, right? Those high energy electrons, as they release their energy, that energy is used to pump hydrogen ions against the concentration gradient. In a sense, you could almost think of like if you had a pump down here at the other end of the dam, and you were pumping the water back into the lake. It's kind of the same thing. And again, like I said, the fact that we have an inner membrane and an outer membrane is important, and this is why. Now you know why. What's happening, even though it's hard to see it in this picture, right? We've got all these hydrogen ions inside the inner membrane. They're getting pumped out, right? And then they're caught. They're trapped there between the inner membrane and the outer membrane. They have nowhere to go. But nature does its thing, right? They want to go back down the concentration gradient, right? This is how things work. You know about diffusion. Things want to go from an area of high concentration to low concentration. And that's where this comes in. There's an enzyme called ATP synthase. And it is the thing that lets the hydrogen ions diffuse back down the concentration gradient, as they want to do, right? Again, it's just nature. Diffusion is happening. After we pump them against the concentration gradient, they just do what they naturally would do, which is diffuse back down the concentration gradient. And to remind you of what you learned previously, this is what we call facilitated diffusion, right? So it's still diffusion, but remember, like I said, sometimes you need a protein for things to go in and out of a membrane. This is the case here too, right? This protein is called ATP synthase. It allows the hydrogen ions to come back through. And in the process, that picture's not from your textbook. I don't know if your textbook talks about this or not, but in the process of doing that, it literally turns the turbine, right? There's a little spinny thing on that enzyme, and that's what produces the ATP. And also notice, I'm sorry, I guess we're done for the day. Last word for attendance, other than asking me a question, will be Columbia. It's in the capital of South Carolina. Sorry, I held you guys up. If I can remember, we'll get out a minute earlier on Wednesday. All right, that. If you guys need grades, just follow me to my office, please.
And another